All right. Well, thank you for clapping before I give the talk, and I'll be interested to see if you clap afterwards. So hopefully. So all right. So I want to ask a question, which is a very simple one for biologists, but a very complicated one for physicists, and that is, what can we learn about life and biology without actually knowing very much about it? All right. And I'm going to, and why that question is particularly important is it's not meant to be an excuse for doing bad science. When I do biology, I work with biologists. We do very, very detailed work understanding the biological mechanisms of the systems that we're looking at. But when you're talking about the origin of life and the early emergence of life, you have such little information to go on. You have to try to tease everything out in a way that is sometimes uncomfortable, but you have to do it. So what I'm going to talk about is something very concrete, the genetic code. And I'm going to talk about some of the things that happened, the major evolutionary transitions that happened in the first billion years of life. And I want to precede the discussion and end the discussion with some quasi-philosophical points about how physicists should be doing biology. It's something that I think about a lot. And you'll see why it's not an empty question, but it's important if we're going to understand practical things in biology, like how to treat diseases. Okay, so this is work with Tommaso Biancalani, who is now at MIT, Farshid Jafarpour, who is now at the University of Pennsylvania, Glenn Sigyan, who is at the University of Wisconsin, and Carl Woese, who unfortunately some of you may have heard of, but is no longer with us. All right, so the question I want to understand is the sorts of evolutionary transitions that life must inevitably pass through. And I use the word inevitably. We want to understand, are there patterns of transitions, patterns of behavior, things that natural progressions that we should expect for evolving systems? And those transitions are inevitable. They're part of the dynamics of evolution. They're not part of the specific details of some particular biological system. So we want to look for universal characteristics of modern cellular life. And the question we want to ask is, can we understand these features from general dynamic principles without necessarily having to worry about all chemical details? It's not saying that they don't matter. It's just we want to try to understand what is important for understanding features that are general, and what features and what is idiosyncratic has to be understood in a different way. So I'm going to focus on two things. I'm going to focus on the canonical genetic code, and if you don't know what that is, I will explain it to you in the talk. And I'm going to talk about chiral symmetry breaking in biology, biological homochirality, which is the fact that if you look at the amino acids and sugars of life, these biopolymers have all one-handedness, one sign of chirality. And there isn't at the moment a good explanation of why that is so. And why is it important? Well, I claim that if you can understand why this is an inevitable feature of all living systems, and we'll talk about why you might think that, then you can use this as a biosignature for life on other planets and other environments. And that's one of the things that we're interested in. Our work is funded by NASA, and we want to understand what are universal signatures of life elsewhere where there are no false positives. Okay, so we're going to see that, in fact, this is something that is not just something we see anecdotally on Earth, but something that we should actually expect. All right, so let's talk about universality. I want to first remind you about universality in physics. So here is an example of universality in physics. So this is from a paper by Nobel laureate Tony Leggett on the amorphous properties of glass. And what you're looking at here is the thermal conductivity of different types of glass. And then if you scale the data in some particular way, the data fall onto one universal curve. And that's an example where it doesn't matter what the material is, it still behaves exactly the same way when written down in the correct units. So that's something that is a surprise. And in fact, it's something that is still not understood. Here is another example. So this is an example that's dear to my heart. This happens near a second-order phase transition. So a second-order phase transition is one where there is no latent heat, and so it happens not by a big jump, as something starts off being liquid and say turns into solid, like ice, water turning into ice. It's not something like that. It's a transition that occurs in a more gradual way, such as, for example, a magnet a bunch of spins, of dipole moments, that as you lower the temperature, gradually more and more of them start to point up in the same direction, either up or down. And as you lower the temperature further and further below some special temperature, more and more of the spins point up in the same direction. So magnetism occurs through a gradual transition, and that's called a second-order phase transition. The magnetization of a magnet, that's what we'll call M, is a function of temperature, but it's also a function of an external magnetic field. If you take a piece of metal and you find it's magnetized, it's because it was exposed to an external magnetic field at some point in its past, like the Earth's magnetic field, for example. So this is a function of two variables, H and T. Now, this is something that you have to know about phase transitions to really understand, but if you are close to the transition at which a material like iron first becomes a magnet, if you take the magnetization and scale it with temperature in a certain way, and you take the temperature and scale it with magnetic field in a certain way that the theory predicts, all the data fall onto one universal curve. And these are data from 5 different magnetic materials, chromium bromide, and so on and so forth. You can read the names here. They're all different materials, and yet they fall on the same curve. And the solid line that goes to those points is the theoretical prediction based on the normalization group. So there's a universal behavior here at this second-order phase transition. And in fact, if we had looked at the liquid-gas transition very close to its critical point, there's corresponding quantities that you can plot that would also fall on this curve. So you get universality not just between magnets, but between different phases of matter, based on symmetry. And this is completely well understood. Okay, so now I want to talk about why it is that we have universality. So let me tell you what is surprising about it. If you look near the critical point of, say, a magnet, you find that certain quantities, thermodynamic quantities, like heat capacity, or how much the system responds to an external magnetic field, the susceptibility, those things go to infinity as you get close to that critical temperature. And the way that they diverge goes as a power law in the temperature minus the critical temperature. Okay, so this is known, and this was explained and understood, and a Nobel Prize was awarded to Ken Wilson for the invention of the method. Now, the thing about it is, when you look at the physics of a magnet, what you find is this. You start off at the lowest level of description with quantum mechanics. Then you have electrons in a solid. Those electrons in a solid are coupled together in some complicated way through the wave functions and the spins and so on. So then you say, let's forget about the electrons. Let's just look at the spins. And now I have a model of quantum spins. That model is still too complicated to solve. So you say, OK, let's make the model classical. So you make the model classical. You say, throw away quantum mechanics. Can't justify it. Just throw it away. You do that. You still have a model that's too hard to solve. So then you say, OK, let's just look at the symmetries of this model, and let's just write down some very simple free energy. Technically, it's called a Landau free energy. So you write that down, and then you compute with that using the normalization group, and your result agrees exactly with experiment. Okay, so this is what's amazing. You have a model that gives precise agreement with experiment, but actually it's a model of a model of a model of a model of a model. Okay, it's a model where you start off with quantum chemistry, then you go to electronic structure, so-called quantum Heisenberg model, the classical Heisenberg model, and eventually Landau theory. Every step along the way here is a step that you cannot look somebody in the eye and justify. They're non-systematic approximations, and yet the result agrees exactly to the best way that we can measure with experiment. That's fantastic. That is the miracle of universality. It's the miracle of how details cannot matter in some situations. Okay, so that's the paradigm. Okay. Now, I want to talk about biology. Now, why would a physicist talk about biology, particularly a physicist like me, who actually, don't tell anyone, I don't actually like biology very much, right? I don't know much about biology, and I want to try to explain to you why it's interesting for a physicist like me, right? Why do we learn new physics when we study biological physics, or is it just insanely complicated soft material science? What are the universal phenomena in biology? Do they reveal anything important? And most importantly, which I'm going to spend a bit of time on at the end, what do we miss by not understanding universal phenomena? Now, any biologist in the audience, like one of my friends is here, will say, well, you're mad. There's no universal phenomenon in biology. Everything is different. It's all exceptions. Everybody knows that. But I'm going to show you that that's not true. And in fact, if we think that, we're going to miss something very important. So let's talk about what I want to call universal biology. Okay. So here's an example of a headline that you might have seen on the internet from a NASA website. Mystery of the missing waves on Titan. Okay, so Titan has oceans, methane. It has an atmosphere. The atmosphere is moving. The atmosphere moving creates waves on the surface of the ocean. There should be interactions between the atmosphere and the ocean. We can calculate that because we know the equations of hydrodynamics. We can calculate how big the waves should be, what their wavelengths should be. Everything about them, we know they should be there. When you go and look, they weren't there. So there was a mystery of the missing waves on Titan. Now, if it kept you up at night, you'd be happy to know that a few months later, this was the next headline. Cassini spies the wind-rippled waves on Titan. It's all right. We found those waves. Everything is there. So it all worked. Okay? What we predicted was observed. Here's a headline that you surely didn't see. Oh, don't tell me I didn't put it in. Oh, I didn't. Oh, well, let's see. Okay. All right, this is what I just said. Okay, next one. Here's the headline. Mystery of the missing life on Titan. Anybody see that headline? And what about this one? Cassini spies long-sought life on Titan. Anybody seen that one? Okay. Actually, this one nearly came true, but not Titan, Enceladus. Okay. Now, why is this one a joke and the other one not a joke? The reason is this. We don't have a theory that predicts the existence of life as a physical phenomenon. So when you say there should be life on Titan, why? What's the basis for that prediction? We don't have that. Nor do we understand what kind of life can arise in different environments. So it's a joke for that reason, and this is an even bigger joke. Now, let me just tell you, when I wrote this, Cassini is a satellite. It's a fantastic satellite. It was a joint European Space Agency NASA mission. It went, excuse me, it went to the distant planets, and it made many, many discoveries. One of the things it discovered was that if you look at the moons of Jupiter and Saturn, Europa and Enceladus, they're far away from the sun. They should be frozen cold, and they have kilometers thick layers of ice on the outside. But inside, they have a global ocean of liquid water. Okay? Who knew? Okay? It's amazing. We didn't know this a few years ago. Now we know it. And how it was discovered was by physics, because those planets move around their orbits, and suppose I give you the question, how do I tell whether an egg is hard-boiled or is liquid? Okay, you could play Russian omelette, is a game we used to play. If I'm ever put up for the Supreme Court, and they ask me, what did you do as an undergraduate? Were you drunk all the time? Yeah, we used to play Russian omelette. Get an egg. 5 eggs are hard-boiled. One of them is not. And do that. Okay. All right. How can you tell whether an egg is hard-boiled? Well, you just spin it, and then you stop it, and then it'll keep spinning again if it's liquid. So the angular momentum keeps going. So the angular momentum of the liquid inside will cause a rotating object to have these little oscillations like that. And Cassini was able to detect those. And that's how we know that underneath the ice is a global ocean of liquid water. Why is it liquid? Because they're so close to this planet that the tidal friction melts the rock and causes tectonic activity and heats it up. Okay? So we know that's true. Now, the other thing, why I say this isn't such a joke anymore, okay? In Enceladus, this little moon of Saturn, which has a global ocean of liquid water, it also has a fountain, a water fountain. There's a crack in the ice at the South Pole, and out of that ice is coming a fountain of water. And we can send Cassini through it, and we did, several times. And we only analyzed the data correctly a few months ago. And what we learn is that the data shows that the water chemistry is not what you would expect from equilibrium water. It's actually non-equilibrium. It's out of equilibrium. It's like the water chemistry that you see at hydrothermal vents on Earth, alkaline hydrothermal vents, which is exactly what you would expect based on what we know about the geochemistry. So we haven't actually detected life because Cassini wasn't instrumented that way, but a future mission could. And it's a very interesting thing. All right. So that's what I wanted to say about Cassini. this field of astrobiology, which is a field that is just now becoming a real precision science due to things like Cassini. All right, now, what I'm interested in is general principles of living systems and not specific carbon chemistry details. So we need a theory of universal biology divorced from idiosyncratic details, and that's going to involve abstractions. We're going to have to take biological systems and then find a way to abstract them. As physicists, we want to make abstractions that are mathematical. Most biologists will say, okay, that's just ridiculous and you're just out of it. But I'll show you that that's not true. And in fact, there's a major abstraction that biologists use all the time and they're very comfortable with. And we're going to get to that in a few minutes. Now, I want to tell you on this slide what we know about the universal biology so far. Okay, so that's it. Okay, so having said that, let's talk about some other field where we have some knowledge of something universal, and that field is universal computation. So let me tell you about universal computation, because the comparison with biology is very interesting. So what do we know about universal computation? Well, what is this thing? What is it? It's a laptop. What does it do? It's a computer, right. Okay, what's this thing? Yeah, you know the answer. Okay, it's cogwheels and springs and levers. It's a Babbage machine. It was on display at the Science Museum in London and in the, just not far from here, the Museum of Computation in Mountain View. Okay, this thing equals that. Okay, this thing and this thing are not shiny chunks of glass, plastic and silicon, nor bunches of cogwheels and springs and levers. They both do exactly the same thing, that there are Turing machines with a von Neumann architecture. And why? This thing, when Babbage designed it in the 19th century, never finished building it, it had conditional branching. And if you work out what this thing can compute with conditional branching, this machine of cogwheels and springs, it is a universal Turing machine. It can do any computation that can be done in this machine. Very slowly, and it would require a machine as big as this building, but you can do it with cogwheels and springs, or you can do it millions of times smaller and millions of times faster with integrated circuits. But the computer is neither one or the other. It's the mathematical construct of the Turing machine with a von Neumann architecture that can be instantiated either in this world or in this world or in this world. This is John von Neumann standing at the Institute for Advanced Study in Princeton in front of a machine, the first computer, the MANIAC, built, one of the first computers I should say, built out of not this and not this, but out of relays and thermionic valves. Okay? So the point is that there's substrate independence. It doesn't matter what it's made of, it's what it can do, and the function is the important thing. So the medium is not the message. Okay, now physicists went into biology. They looked, Schrödinger and Delbruck wanted to look for new physical laws, but they thought that they could see corrections to quantum mechanics. Today we don't think that's very plausible, but the question that we really want to understand, or I want to understand, is why life exists at all. How does matter self-organize hierarchically to create replicating and evolvable structures? How do molecules come to life? So I want to talk then about what it means to understand a scientific problem. And what it means to understand it is we understand why the thing exists in the first place, and not just a specific instantiation. Now, what is universal about all living systems is that they are the products of evolution. And what is evolution? But it's systems that can reprogram themselves. So if you take one thing away from this course, this is what biology is. The program is the data is the program. Okay? So the program literally acts on itself, it's self-referential, and as it changes the program, it's changing the data, that changes the program, and so you get this endless self-referential chain. Now, the first person to realize this was this guy, Niels Barichelli. He also worked at the Institute for Advanced Study in Princeton when von Neumann was building his computer. Von Neumann wanted to compute what happens for the hydrodynamics of thermonuclear explosions. He was funded by the military to do that. At night time, when von Neumann and his engineers had gone home, this guy, Niels Barichelli, made one of the first computer viruses, and he looked at how they interact. And this printout here, which George Dyson gave me, is some representation of the time evolution of a bunch of organisms living inside a computer, fighting for memory and resources in the computer, and evolving and replicating and so on. So living systems self-program, and that's very different from physical systems, which obey a fixed or determined equation of motion. And what that means is that the future state of an evolvable system is very difficult to compute. And I'm going to talk about this at the end of the talk. The real question that you want to understand is it possible to even, in principle, control evolving systems? And why this is a problem is because when you look at at medicine and biology applications in medicine, the big problem we have is trying to understand how to combat bacterial infections, how to combat viruses, how to combat herbicide resistance, how to combat insecticide resistance, how to make chemotherapy work and not have tumors evolve so that the chemotherapy doesn't work anymore. All of our attempts to intervene in biological systems end up failing because we don't understand this fundamental question. All right, so let me move on to an example of a successful abstraction in biology. And that abstraction is the genetic code. So let me tell you about what the genetic code is. So here's Biology 101. So you have DNA, which is where your 3 billion base pairs long that encodes your cells. So DNA is built from 4 nucleotide bases, A, C, Gs, and Ts. And there's a machine called RNA polymerase that goes along DNA and spits out a thing called messenger RNA, which is another nucleic acid also built from 4 nucleotide bases. This is kind of like this. Suppose I wanted to fix the projection system in this lecture room. And it went wrong. It's working fine, but let's suppose we wanted to fix it. And so the engineer comes along to fix it, and he says, give me the plans to fix this. And so you give her the entire plans, the wiring diagram, the plumbing, and everything for the whole University of California, Riverside. So she's going to say, well, that's great, thanks very much, but I just need to Xerox off, copy off a few pages, which just is the wiring diagram for this lecture room. Okay. Biology does the same thing. Your DNA is like the whole plans for the entire university. The RNA, the messenger RNA, is like just the specific bit for this particular room. In this case, it's for a particular gene. Now, what do genes do? Genes code for proteins. Proteins are built from 20 amino acids. So what happens is another complicated machine called a ribosome goes along here, reads the message on this messenger RNA, decodes it, figures out what amino acid to stick onto the next at the end of the protein, and the proteins get built in that way. And this is a picture of it, the ribosome, this big machine, moving along, reading triplets of nucleic acids on the messenger RNA, deciding what amino acid that is, taking the amino acid, putting it on, and sticking it onto the emergent protein. That process is called translation. Now, what are these codons? You can see these codons here, AUG, ACA, GGG. They're all in triplets. Why? Because you have 20 amino acids. You have an alphabet of 4. So if you had 2 codons, it would be 4 times 4 possibilities. That's 16. 16 is less than 20. It's not enough to code for 20 amino acids. So you need at least 3 of these things in an alphabet of 4. So you've got 4 times 4 times 4. That's 64 possibilities of messages that you can produce, which correspond to the code that corresponds to the 20 amino acids. So then what you've got then is a genetic code as a map used by the ribosome to translate a message from triplets of 4 bases into the 20 amino acids of life. It is a real code. It's like a code book. It's not your DNA sequence. That's not the genetic code, even though the newspapers often say it is. Okay, now, who was profoundly interested in the ribosome was this gentleman, Carl Woese, sitting on his desk in Urbana, front page of the New York Times, November the 3rd, 1977. Why was Carl Woese on the front page of the New York Times? Well, the New York Times said incorrectly that he discovered a form of life that predates higher organisms. What he had actually done was he was a physicist originally. What he had done was he set out to map out the evolutionary history of life on Earth. And he did that. And the way he did it was he said, look, every cell has to have ribosomes to make proteins. So if I just look at some of the sequence that codes for the ribosome itself, and he chose a particular part of it, so-called 16 or 18S ribosomal RNA. But it doesn't matter what it's called. He chose a particular bit of it and then said every organism has to have this in it. It has to make the proteins. It has to have this bit. Let's look at how that very important part of the machinery changed over evolutionary time between organisms. And then from the way it changed, I can then see how to put together the tree of life. And he did that, and this is what he got. Okay? So here's the tree of life that Carl Woese developed. This is shown in an article from Norm Pace. And you can see here is a bacteria. Here are eukaryotes, the cells with nucleuses that you learned about in high school. There's you. And everything else on here is essentially microbial. This is the thing that he discovered, a third domain of life called the archaea, which nobody knew existed before. They look under a microscope like bacteria, but their evolutionary history is completely different. Now, what I'm interested in is not you or the bacteria and the archaea, but what happened right here where the tree started. That's called the root. That corresponds to what? The origin of life? No, not the origin of life. Why? Because anything you see on this tree, or anything you infer from this tree, had to have ribosomes, had to have a system of translation, had to have a complicated cell architecture. Anything you see on this tree, then, is not the beginning of life. This is a very sophisticated form of life, but maybe the first ancestor of all organisms on Earth. This tree shows us that all of life is related. And this organism or community here is called the last universal common ancestor. Now, here's the thing. When you go and find and see what organisms are near there, as Woese and Fox did in their 1977 paper, what you find is that these organisms are blue-green algae, what we call today cyanobacteria. The time available to form them is very short. Why? Because we know how old those organisms are. They thought they were 3.1 billion years old when they wrote this paper. Actually, today, we know they're more like 3.8 billion years. The Earth is how old? 4.5, 4.6 billion years old. Okay, so 4.6 minus 3.8 is a number less than a billion years. So that meant that life had to have gone from nothing to essentially the complexity of the modern cell in way less than a billion years. Okay, now, 3.8 billion years ago, you had the last universal common ancestor. After that, nothing much happened evolutionarily speaking. Okay, well, maybe you had a few dinosaurs and humans and things like this and plants, all right? But basically, the architecture of the cell didn't change that much. Okay, so how did it go so fast then? And what Woese and Fox argued in another theoretical paper that year, they said, look, evolution can either just go faster. There could have just been much faster rate of mutation, or it could be that evolution happened through a different mechanism than just mutations, which was what their prejudice was. Okay? So they felt it more reasonable to assume it was the mode and not the tempo of evolution that changed. And what could that other mode be? So here's what that other mode is. It's called horizontal gene transfer. So some of you know I teach a graduate class in statistical mechanics at the University of Illinois, and in particular a class on renormalization group theory and phase transitions. Okay, very complicated mathematics and field theory and stuff like that. Okay, now suppose we could do it this way. Suppose there was, let's suppose, a gene that enables you to do complicated Feynman diagram integrals in 4 minus epsilon dimensions. Okay, there isn't, but let's pretend there is. Okay, because the newspapers sometimes say there's a gene for cancer, a gene for alcoholism, a gene for, why not theoretical physics? Suppose there were genes for that. And I could just take that DNA and just pop it out and throw it out to the audience. And you could take that gene in and then, bingo, you could learn how to do those calculations without having to sit through a course. That's science fiction. That doesn't happen. Okay? Nobody teaches theoretical physics that way, or should. But if you were bacteria, that can be done and is done all the time. And it's called horizontal gene transfer. You take a gene from you, and it's horizontal because I send it to you who's not related to me. If you send it to your children and your grandchildren and so on, that's called vertical transmission. So horizontal transmission of genes is to somebody you're not related to. Bacteria do this, and they do it through 3 different ways. They can do it through picking up DNA from the environment. They can do it by just meeting each other and transferring little plasmids, or they can do it by a virus attacks one organism, replicates itself 100 times, stuffs its capsid full of DNA, Sometimes it'll get the wrong DNA, it'll get the DNA from the host organism, fly out, infect another bacterial cell, and transfer some of the DNA from this cell into that one. And that happens all the time. And that's called transduction. So horizontal gene transfer happens, and the question that a statistical mechanician like me asks is, if microbes can do this on a pairwise basis, what happens when the whole community does it? Now, one answer is a tragedy, a health crisis. So you may know that antibiotics don't work anymore. They're starting to not work. A lot of diseases are now at the antibiotic of last resource. There's some diseases, I think gonorrhea and TB, where now there's cases where there's no known antibiotic that can treat them. And why is that happening? It's because right now the genes in your gut, the bacteria in your gut are busy trading genes just like this. And bacteria have genes that promote resistance to antibiotics. Antibiotics are simply drugs that do things like disrupt the membranes or stop translation and things like this. Bacteria have used them for billions of years to wage war against each other, and some of them have developed immunity to them, and they share them around. So the evolutionary timescale for bacteria globally is not millions of years. It's like 5 years, 10 years. And that's why we have a global health crisis now, which the World Health Organization calls our most severe one. Here's something really wonderful, which I want to very quickly tell you about. These are viruses that infect a cyanobacteria. These are cyanophages. And what's interesting about these is these are phages, these are viruses that infect bacteria. But the phage, if you look at its DNA, it has photosynthesis genes in it, photosystem II genes. So you're saying, wait a minute, why does a virus need photosynthesis genes? Well, you can know why the cyanobacteria needs photosynthesis genes, because they live in the ocean and they make their energy from sunlight. So, okay, bacteria has photosynthesis genes, that's fine. That's where plants got them from originally. But why would a virus need them? Well, here's the thing. You look at the molecular phylogeny, you look at the evolutionary history of the genes, these photosystem II genes. And what you see here, I'm not going to explain to you how to read this chart, but what you see is that those photosynthesis genes have been transferred back and forth multiple times between the bacteria and the virus. The bacteria has taken a photosynthesis gene, it's been transferred to a virus. The virus has transferred it to other viruses. The virus has transferred it to other bacterial cells, and it goes back and forth between the bacteria and the virus population. So what we now understand from the work of primarily Penny Chisholm and her group is that what's happening here is that imagine that you're a virus and you infect a cell. So here's your problem. You want to make as many baby viruses as possible. You go in, you start hijacking the cell's machinery to make baby viruses. The cell, of course, is dying while you're doing that. And maybe the cell lives long enough that you can take out enough material to make, let's say, 100 baby viruses. Suppose you could put the cell on life support. You could put the cell on like a life support system so that it lives longer. And then it will produce maybe 110 baby viruses rather than 100. Any virus that carries a life support system machine around with it will have an evolutionary advantage over one that doesn't. Or it can go in and accelerate the metabolic activity of the cell and make the baby viruses much faster. So a virus that has that ability would have an advantage. And so it's advantageous for the virus to carry photosynthesis genes in its back pocket so it can keep the cell alive longer and increase what's called the burst size of the virus. So what you end up happening then, and this is the sort of conclusion of the Chisholm Group's very sober paper, is something that sounds a bit like science fiction. Host-like genes acquired by phage undergo a period of diversification in phage genomes that act as a genetic reservoir for their hosts. And this enables them to both the host and the phage to evolve better photosynthesis genes. In work that we've done, which we haven't yet published, we've shown how this mechanism enabled the evolution of a low-light adapted strain of Prochlorococcus deep in the ocean to be able to evolve to be able to live very close to the surface, where there's a much higher intensity of light and where the wavelength of light is different. And they were able to do this rapid evolution through this horizontal gene transfer. So the take-home message is that the cloud was basically invented 3 or 4 billion years ago by microbes. Okay, now here is the genetic code. So you have triplets of bases. And you read it like this: U, then in the next column, U, and then one of these columns here. So U, that codes for phenylalanine, and so on and so forth. These are the 20 amino acids that life uses, and the colors tell you the most hydrophobic ones. These blue ones here are the most hydrophilic ones. And this structure in the genetic code table was realized by Carl Woese back in the 1960s. This is an abstraction. You can wear it on a T-shirt. It tells you all the machinery, all the biochemistry that is going on when the ribosome is moving through the mRNA and spitting out the proteins, and the synthetases are charging the tRNAs and all sorts of stuff like that. All this complicated biochemistry is just encapsulated by, okay, it doesn't matter what happens, it just happens, and this is the result. That's an abstraction. Okay. So what Carl Woese did was he measured this hydrophobicity and hydrophilicity, and he discovered that it's small over here and a large number over here. Now, why is that important? I'm going to tell you something about the genetic code that you didn't know. You know that the genetic code is the same, pretty much, with minor modifications, for all organisms on the planet. That's how we can do genetic engineering. That's how you can make things light up with green fluorescent protein. You can just take a gene from a jellyfish and stick it in a mouse, and it'll glow green. Okay? So the genetic code is universal. It's the same for everything. Okay? But what you probably didn't know is that it's not just universal, it's nearly optimal in minimizing errors. So let's suppose you had the following homework assignment. You wanted to know, I want to do a little bit of intelligent design. I want to design a genetic code that is the best possible one so that I won't get errors. Now, what do I mean by errors? Well, let's suppose that there's a mutation, or let's suppose that you just simply misread as you go along. You're going to get the wrong amino acid. Can you structure the genetic code in such a way that the amino acid you get will be pretty close to the one that you should have got, perhaps even close enough that it won't make a difference? The answer is yes. You can construct such a code, and the code that is the one that has that property is the genetic code, the one we actually have. How do we know this? We've only got one genetic code. We can't take a bunch of organisms and say, how good is this one at minimizing errors, how good is this one, how good is this one, because everything has the same genetic code. But we can do a computation. So what you do is you take the actual genetic code, and then you permute the labels around. So how many ways are there to do that? Well, I've got 20 amino acids, so I've got 20 factorial ways, which is about 10 to the 18. I can't enumerate 10 to the 18 codes, but I can enumerate, let's say, 1 million of them in a computer. Okay, so I can make lots of genetic codes in the computer. All right, then I have to decide how good they are at minimizing errors. And I'm not going to go into detail how you do that. There is a way that you can look, basically, for those of you who want to think about it in this way, I basically look at the Laplacian operator on the space of the amino acids in the genetic code, and I try to compute that and minimize it. Okay? So there's a way you can do this. You can basically compute the sums of the squares of the differences of the polar requirement numbers. And you can do this, and you can get a number that tells you how good a genetic code is at minimizing error. All right, so now I can take a code, and I have a figure of merit that says how good it is. Next thing, now do this for every possible genetic code that I can construct in a computer or a good representative sample. Let's have a look at what the output could be. The first output could be this. This is the naive expectation, which is what Francis Crick thought would happen. He called it the frozen accident hypothesis. The idea is the last universal common ancestor was some organism. It had some genetic code. We have to have the same genetic code as that organism, because if you made a change to the genetic code, then you'd have the wrong proteins. If you had the wrong proteins, you'd die. So the genetic code can't evolve. It's frozen. It's a frozen, and it's an accident because it's just whatever organism happened to be our ancestor. So that's the frozen accident hypothesis. How do you test it? Well, you generate a bunch of codes at random. Let's suppose you get this bell-shaped curve where this is the frequency at which you get a particular score that tells you how good that code is at minimizing error. So you get that frequency distribution, and then you ask, where is the actual genetic code? If it's there, that red line right in the middle, it says, okay, our genetic code is just what you would guess at random, nothing special. Suppose, on the other hand, you got this. Here's what you would get from random codes, and here is what the actual genetic code is. Smaller is better. That says it's not what you would get at random. Something actually happened to make it like that. Something evolved. So here's the answer. So this was a paper by Freeland and Hurst in 1998 called "The Genetic Code is 1 in a Million." There's a million genetic codes synthesized here, and there's technical details that, of course, I'm glossing over. This is what you get at random. This is what the actual genetic code is. In a million codes, not one is better than the actual genetic code. We've done this calculation in many different ways, and this number is actually much better than the number that they give, but it's still the same thing. So the genetic code had to evolve. But it can't evolve because of Francis Crick's frozen accident idea. So we figured out a way to get around that. We figured out how the genetic code actually did evolve, and I want to tell you about that. I'm not going to show you the mathematics, and I'm not going to tell you all the details of how we did this. I'm going to walk you through a thought experiment that will allow you to see how a genetic code can evolve. So this is from a paper that we wrote back in 2006. So the basic idea of how a code can evolve, I want to explain it to you by talking through a thought experiment between people who are experts in codes. Now, who are the experts in codes? Who? You are? Okay. All right, you are, but what group of people are experts in codes? Okay, I know why you say that, but which class of people? Cryptographers, spies, National Security Agency, right? Okay, these people. Okay, okay. All the people who are laughing are people who are definitely over 40 or something like this, because they recognize these people from bad 1960s TV shows and so on. Okay, all right. So let's suppose a spy wants to communicate through a code book with another member of their learned fraternity. So we're going to talk about what can go wrong with codes. So here's the idea. Let's start off with a message. So Emma Peel has a message. There's the message. She sends it to Austin Powers, takes the code book, and interprets it. And indeed, that is the correct interpretation of this code. So everything works fine. And what you've got here is you've got a single message. You put it through a code book, you get a single unencrypted message. Everything is fine. Now, the problem is that this guy is a very careless and accident-prone individual. He has many other obnoxious characteristics. And the code book gets broken. The code book is now, half of it's been lost, and now the message makes no sense to him, although it might make sense to you. Okay? So what you learn from this is that if you change the code, then the message gets garbled. So this is the frozen accident hypothesis of Crick in action. Suppose now you have something different. You have a code book that is stochastic. It's a code book that doesn't produce one message, it produces a whole ensemble of messages. And you look through them and you say, what the heck, that doesn't make any sense, that doesn't make any sense. Ah, that's what it must be. And you can work out the probability. In this case, he works out the probability of this being the message is the one. So a stochastic code book, one that is not a one-to-one mapping, but produces a distribution, is one that could be immune to errors. Now, let's think about how life could have evolved early on. Early on, life was, sort of like, well, think about automobiles, okay? Today we have nice cars like BMWs and Toyotas and things like this. But, hundreds of years ago when people started to invent automobiles, they were really, really terrible, right? They would fall apart, they couldn't go very fast, they didn't have Bluetooth and stuff like this. They really weren't worth driving, okay? They were terrible. But they did have the following feature. If I take my car that I've built by myself, perhaps, and I go round to my friend, and the wheel falls off my car, or something goes wrong with it, I can just rummage around in his basement, find a spare part, and put it into my car, and it will still work. It'll still work in one of those old automobiles. Let's suppose that I don't have a BMW, but suppose I did. I drove my BMW around to your house, and the wheel fell off it, and you said, well, don't worry about that. Here's a part from my Toyota. Well, forget it. That's not going to work. It doesn't work because the automobiles are too complex. So now think about early life. Early life was life that could just about barely make a living, could barely function. If the protein didn't fold in exactly the right way, it didn't matter because it didn't have to do very much. It wasn't high performance. The proteins weren't built to 6 sigma tolerance. But fast forward, few billion years, now it really matters. If you get a misfolded protein in your brain, you get all sorts of terrible diseases like dementia and things like this. So it really, really matters today, but it wouldn't have mattered several billion years ago. So then the idea is to make a dynamical system, which says how does the complexity of life co-evolve along with the code that codes for it. Now, let me show you how that works mathematically. So the idea is then that the code and the organisms, as they evolve and become more complex and need greater accuracy, the code itself evolves along with that. And it can evolve because it's a code that has a distribution, and so you can always find what's going on. OK, so here's an example of how selection pressure can influence code dynamics. So you might say, wait a minute, this can't work, because selection operates on the phenotype. It's not operating on the code. Well, it's not true. It does operate on the code. So let me show you how. So I'm a physicist, but I often collaborate with biologists, and we write many proposals together, and some of them we get. Okay? When I started, I always wanted to write my proposals in TeX, in LaTeX. My microbiology friends wanted to write them in Microsoft Word. And so we'd bicker back and forth about, I want to send you some TeX, they want to send me some Word. I use Unix, so I can't even run Word. So things weren't very good, okay? And maybe my colleague down the hallway, who's a bit smarter than me, just did it in Word, sent in a proposal to NSF that wasn't really as good as the one I would have done, but it gets funded and mine doesn't. Why? Because I never even got to deliver my proposal because I was too busy bickering with my colleagues about what platform we should use. So what you learn is that you can have selection based on the code. And of course, today, I routinely write proposals with biologists in Word, and I just deal with it. OK, so here's another thing. Here's two other computer analogies. There's some platforms. This is Bill Gates with his Microsoft Windows operating system. Here's Steve Jobs with his operating system for the Apple. Hey, they're both visionary technologists and so on. But the fact of the matter is that 95% of the world's computers run Windows, and a few percent run the Macintosh. I happen to have a Windows computer. Some of you have Apple computers. I have a Windows computer, and the reason I have a Windows computer is because they're better. They're more reliable, they work. That's an inopportune time to have a blue screen of death. OK, so none of that happens. So why is it true that the Microsoft Word, Microsoft's operating system, is gained global acceptance, whereas the Apple one didn't? Well, it's because you have a greater pool of innovations available to you if you use Windows. It's maybe not so much true now, for reasons I'll say in a minute, but it certainly was the case maybe 10 years or so ago, that if you wanted to get a program to do X, Y, Z, somebody would have written it for the Windows platform, but they wouldn't have written it yet for the Macintosh platform. And that's still true today. For example, in the early days of smartphones, somebody has a smartphone here. In the early days of smartphones, the big competitive advantage of the iPhone was what? The App Store, right? You have 1 million apps on the App Store, and only a few 100,000 on the Google one. Okay, now that's changed. And in fact, Apple realized this. Steve Jobs changed the chip in Macintosh computers to be the same Intel chip that the Windows computers use. So you can now run Windows on Apple computers. Okay, so people understood this. So that shows you that that codes can evolve, and that the most important thing is that communities that have access to a greater pool of innovations have a competitive advantage. In the case of living systems, you're trying to invade a new ecosystem. You're trying to compete with other organisms. If you have access to genetic innovations, like the example I showed you before, the photosynthesis genes, you have a competitive advantage, and you'll win. So what we did was we did a computer experiment, kind of like the one that Niels Barichelli did back at the Institute for Advanced Study, back in the days of von Neumann. He made computer viruses, and we made computer viruses. Today we call them digital life or digital organisms. And we made an asexual population of evolving, reproducing digital organisms that live inside a computer. But our ones were better than his because they didn't just have a fitness and they just reproduced and so on. Their fitness was a function of their phenotype. they had proteins that were obtained by translating a synthetic genome from a genetic code with errors, and the individual reproduction rate is a function of fitness. So basically, these organisms, these digital organisms with different genetic codes, can evolve in an ecosystem and compete. And the question we were trying to ask is this. If I start off with a community of digital organisms that emulate what we see in living systems, will we be able to evolve a unique and optimal genetic code? So we tried it. So here is look at the red curves for the moment. So here is what happens. This is the quality of the code. Smaller is better. This tells you how good the code is at minimizing errors as a function of time. You run the simulation, it goes down this trajectory here, gets to this point, and then just goes along this line like this, and nothing ever changes. You run it again, goes around this one, let's say, and then comes along here. You do it again, you get to here. And if you plot the probability distribution of what you would get at random and compare it with these, you find that these are pretty much indistinguishable from what you would get at random. This picture is exactly what we saw before with the frozen accident hypothesis. The codes evolve a little bit and then stop evolving. Okay? So the experiment was a failure. But then we switched on horizontal gene transfer. We allowed the organisms to swap genes by horizontal gene transfer. Today's organisms do not swap genes that are fundamental to the cell's ability to do translation. They swap antibiotic resistance genes, other genes that are not so important, but not fundamental ones. But early on, let's suppose that they can do that. Then something very dramatic happens. The simulations always converge to some value over here, very different from what you would get at random. And so what you learn from this is horizontal gene transfer leads to optimality. Then you count how many organisms, how many codes you have in your system, and again the same thing is true. Without horizontal gene transfer, you get nothing that's different from random. With horizontal gene transfer, presumably, by the way, through endosymbiosis, you get a unique genetic code. So we learned that horizontal gene transfer leads to universality. So what I've just told you is that there was and the other thing I want to emphasize, let me just go back here, is I can't prove that if I didn't wait not 300 time steps, but 10 billion time steps, that this thing wouldn't evolve to here. Maybe it would, but it certainly would take a lot longer. So you can see that this horizontal gene transfer process is much faster. So what I've just showed you is that if I start up with communities of organisms competing and having many different genetic codes, what will eventually happen is that you will end up with a network of life due to the horizontal gene transfer, and then eventually they will make and that will happen very fast, and you'll end up with a unique optimal genetic code. I'm not going to tell you how you transition from this collective era to the era of individual species and organisms, and you get today's slow evolution of a tree of life rather than a network of life. But we were able to understand that too. I'm going to skip homochirality, and I'm going to talk about this. I didn't want to talk about anyway. What I want to talk about is the following question. Let me get on here. What do we miss by not understanding universal phenomena? So I've shown you that there is a universal genetic code, and I've shown you that we can understand it by ludicrously simple models that miss out a lot of the chemistry and so on, but abstract the important things. Okay, now let me explain to you why I think this is important to do. And I want to go back to physics, and I want to go back to superconductivity. So here is the timeline for the discoveries of superconductivity. Superconductivity was discovered in 1911, the discovery of zero resistance. Then in 1933, the discovery of the Meissner-Ochsenfeld effect, which is the expulsion of magnetic field. Then in 1955, the discovery of the Abrikosov vortex lattice. And then finally in 1962, the discovery of Josephson effects. The question is this: why did it take so long to make these discoveries? Were people just not smart enough then? No, same as now, okay? But it took 50 years to go from here to here. In fact, even here, 1957 at the University of Illinois, the theory of superconductivity, so-called theory of superconductivity, the Bardeen-Cooper-Schrieffer theory, was developed. Even with that theory, you couldn't explain this, you couldn't explain this, at least not, it didn't predict it, and this was difficult to explain, although eventually was sorted out. Okay? So even knowing the theory that a Nobel Prize was given for this phenomenon still didn't explain this. The thing is this, when I teach this on the basis of symmetry. I teach superconductivity from the point of view of something called off-diagonal long-range order, a kind of quantum order, like magnetic order, but in quantum mechanics. And you have long-range order, off-diagonal long-range order of a charged condensate, electrons in Cooper pairs, coupled to an abelian gauge field, which is electromagnetism. It's a few lines of algebra to derive all of these things. But it wasn't done for 50 years. So let's talk about levels of description then. I want to now tell you what we lose by not thinking about phenomena in an abstract way. So here's superconductivity, and here's different ways to think about it. I can think at the lowest level about the atoms and the quantum chemistry and the material science. I can talk at some intermediate level about the BCS theory for interacting Cooper pairs, interacting electrons. This is a theory which is also a ludicrously simple theory. It says, forget about atoms, forget about phonons, forget about band structure. The only thing you need to know is that electrons like to pair up, and when they pair up, they do it in time-reversed states or spin-reversed states or whatever. And that's what they worked out the wave function in that case, and it works very well. And then the top level of diagonal long-range order, electricity and magnetism, we'll say Ginzburg-Landau theory. This is the theory, the symmetry theory. In biology, you start off with atoms and molecules. Then you look at an intermediate level of description. So, okay, let me give you some examples. The elasticity of DNA, phase transitions for liquid-liquid intracellular complexes, a very hot topic now in cell biology, and many other things. And then you look at the top level, what is the thing that is common to all biological systems? And that is that they evolve, and you look at the dynamics of evolving systems. We talked about that. We talked about the idea that the program is the data, and the data is the program. So those are different levels of description. What are they good for? They answer different questions. Superconductivity, quantum chemistry and material science answers the question, how does a specific material instantiate the BCS mechanism? How do the electron-phonon interactions happen in a real particular material like yttrium, barium, copper oxide? BCS theory answers the question, what is the basic mechanism for weakly coupled Cooper pairs to form a superconducting condensate? And this level of description, off-diagonal long-range order and electromagnetism, answers the question, why does the phenomenon of superconductivity exist? If we go to biology, here's the levels of description, atoms and molecules. How do specific biopolymers interact, fold, and undergo template-directed synthesis? It answers that question. The elasticity theory for DNA, phase transitions for liquid intracellular complexes, and so on and so forth, answer the question of what goes on inside a cell. What are the basic functional cellular processes? And the dynamics of evolving systems, if we could understand it, would answer the question of why does the phenomenon of life exist at all? Why do you get dynamical systems that can evolve and rewrite themselves and generate the growth of open-ended growth of complexity? The levels of description are equivalent to levels of universality. So in superconductivity, if you have the level of description of quantum chemistry, it describes all materials which are in some specific group. If you have this level of description, the BCS theory, it describes weak coupling superconductors. It doesn't describe lead very well, somewhat, but not well. It certainly doesn't describe the high-temperature superconductors. And this level of description describes all superconductors. Again, if we look at biology, atoms and molecules, that level of description describes specific biopolymers. This level, the physics of particular subcellular components, and this one, all of life. Now, what do we get when we have that level of description? If we have the Ginzburg-Landau theory level of description, it applies to all superconductors. If you don't have it, which is what we didn't have until the late 1950s, early 1960s, we couldn't predict all of those phenomena that we saw, the Meissner effect, the Josephson effect, the vortex lattice, and so on. We couldn't predict them. We couldn't predict how a superconductor would respond to electromagnetic fields. Once you have this top level of description, then you can predict all of these things very easily. That level of description of existential level really has an important effect. It allows you to understand response. Similarly, in biology, we have the dynamics of evolving systems. if we don't have it, if we don't have this universal level of description, we aren't able to predict the response to selective perturbations, antibiotics, insecticides, herbicides, chemotherapy, and so on, the things I talked about before. So in superconductors, by regarding superconductors as collections of atoms, we miss the emergent laws that act at the system scale and govern the large-scale response to electromagnetic fields. We know how to solve that problem. For biological systems, by regarding biology as complicated physical systems, we're missing the emergent laws that act at the system scale and govern the large-scale response to control perturbations. We don't know the answer to that problem yet. And if we don't think about this level, however many new drug targets we find by clever bioinformatics, however many new poisons we think up for chemotherapy, it won't affect the result that cancer tumors will evolve. All right, so this point of view of biology was written down in 2 review articles, one in the Annual Reviews of Condensed Matter Physics, a more recent one, Universal Biology and the Statistical Mechanics of Early Life, that came out in the Phil Trans Royal Society last year. So the questions I asked at the beginning, do we learn new physics when we study biological physics? The answer is absolutely yes. It's new physics. We learn about large fluctuations. I didn't talk about that too much. We talk about self-organization into evolvable, modular, self-programming structures. We learn about new physics. We don't really fully understand all the questions yet, but it's a new arena for physics, for physical scientists. What are the universal phenomena in biology? I didn't go into all of those. There's another talk I have where I go through those and show you that there's many phenomena that are universal, not just the genetic code. Do they reveal anything important? Yes, they do. And what do we miss by not understanding the universal phenomena? We miss the ability to respond, the ability to understand the response and control of biological systems. All right, so anyway, what I showed you was, I didn't tell you about homochirality. I told you about horizontal gene transfer driving the evolution of the genetic code. And I told you about the phase diagram of life. And lastly, I want to end with this quote from Stan Ulam. So people always ask, what can physics do for biology? Can we make a better microscope, a better computational tool, better mathematical tools? But the real interesting question is turn it on its head. Ask what biology can do for physics. And I think I've shown you that there's a wonderful arena of physics that is illuminated by studying biological systems, even for people who come from condensed matter physics and so on. There's plenty of really interesting physics that expands our horizons. And so it's not just what we do for biology, it's also what biology does for physics. Thank you very much.
We have some time for questions or comments.
Would you expect, if you were to find life on Titan or someplace like that you find the same universal genetic code?
I don't think you'd necessarily find the same universal genetic code, but it would have evolved through the same principles, is what I believe.
And why is there only one?
You mean on Earth?
Well, any place. Would you expect there to be only one? That one
Be-
or could you wind up with 2?
No, I think, well, we actually ended up with about 23.
Okay.
So most of them are pretty much the same. They differ slightly by stop codons. Most of the variants are mitochondrial genetic codes. They're not nuclear genetic codes. And there's no law that says it has to be the best one, just very, very good and optimal. So what I would say is you're not going to get a system where there's going to be a million competing genetic codes. There's going to be one, or maybe 2 or something like that. But that's like asking, why are there 3 domains of life? I don't know the answer to 3 domains of life, but I do know that it's not going to be a million domains of life. And in unpublished work, we have some speculations about how that can be. So I think the answer is, when we talk about evolution, we're talking about the process. Is the process universal? The outcome might not be exactly the same. Okay, does that answer your question? Okay.
So, I mean, I think, I'm a physicist, so I'm going to say something about biology that's going to be very bad. But I think in biology there are many, many cases where there are kind of regulating mechanisms, right? Where you have some checks that feedback and use of processes to then maybe, say, reproduce DNA, right? So one of the things you were saying about the code is it saying that it's basically developed a structure that makes that more efficient or less active and more passive? Is there some connection to this kind of error correction and these kind of feedback mechanisms that we know exist? Like, could you imagine
So
an ultimate code that doesn't require any active feedback and it's all just structured in a way?
So in the computer experiments that we did, we didn't have things like proofreading, kinetic proofreading, and other things, all of the other complexities. We simply worked at the level of the abstraction. So what you're talking about are things the machine things hidden inside the machine that definitely make it better, for sure. I mean, you're talking about how the thing is actually instantiated, and real biology does have all of those regulatory phenomena.
I mean, but does it have the implication that there is kind of even without those mechanisms, there's kind of a best structure that would be
Yes.
roughly there, and then
Yes, because those structures, all they do is instantiate that structure. And you can ask, are there any universal properties of these control structures? So I think there is. If you look biologically, what you see often is that the regulation that occurs is master regulators which have negative controls. That is, instead of having some switch that does something or other locally, you have things under the control of a regulator, and it just lives there and stops things happening. And as soon as you turn that thing off, all hell breaks loose. Those things start to happen again. So there seems to be that and many other, or several other motifs of regulation that people notice and see as regularities because they work and have been reinvented or transmitted multiple times. You've had years to ask me questions. I'm going to take somebody else before you run out of time. You had a question.
So, early on in life, there are only a very small number of species. Imagine just one species. Did like horizontal gene transfer that work too, or it just came out after when there were more species? And if that was the case, then how did we have more than one species?
I don't know that the premise of your question is correct. So I don't know that early on there was just one species or something like that. I don't think that's true. Some people argue that, for example, a very extreme example of that is the RNA world. The idea that life started with RNA molecules that did everything and then gradually made a transition to the current world of DNA. I don't find that very plausible. For a philosophical, excuse me, I'm losing my voice here. Yeah. Excuse me. I don't find it very plausible because I think that biology inherently involves co-evolutionary processes. And so I think that, let's suppose you say, well, what's your favorite theory? The alkaline hydrothermal vent theory for the origin of life. So there you've got lots and lots of metabolites that are being thrown up by alkaline hydrothermal vents. You've got chemical evolution going on, producing many different types of organisms. There's no reason to believe it wouldn't be diverse. So that's my answer to your question. Okay, Tom had a question.
There are examples of organisms and organelles using alternate genetic codes.
Yes. Mitochondria, for example.
I believe there's also a few where the nuclear genome uses.
There is one. Yes.
So I just wonder if those codes are sufficiently distinct that you can...
No, they're more or less the same. They may delete a stop codon or something like that. Mitochondria, they are a symbiont in the cell, and so they get actually most of their proteins from the nucleus, not from their own internally. So then the code is basically doing nothing. So if it evolves off in some neutral way, it doesn't make any difference. That's the reason why most of the variant codes are mitochondrial and not nuclear. Yeah.
You described your numerical experiments to test out how the codes evolve. Does the Earth's evolutionary history even have enough time to accurately sample the whole space of codes?
It doesn't have to sample the whole space, okay? Because it's a dynamical system. Okay, this is like so. There was a problem. There's a paradox called Levinthal's paradox in protein folding, which says, wait, here's a protein. It's going to fold into a very precise configuration, but wait, there's 10 to the 300 different configurations it could fold into, it would take 1,000,000 times the lifetime of the universe for a protein to be able to fold. How could it possibly sample them? Okay? That's not how a protein folds. A protein folds, it doesn't sample every possible configuration and then say, okay, I'm going to choose the one that's got the lowest free energy. The protein folds through a dynamical process of expulsion of water that just makes it go like this. And that's how protein folds. So it's a dynamical, almost deterministic process. So you don't have to go and sample all these different configurations and see what's going to happen. So it's a very common misconception. Now, the question I don't know the answer to is this: can I put numbers into this scenario? Okay, can I say yes, if you do this, life can evolve the current level of genetic code in 800 million years? Okay, I can't do that. I have no idea how to put in the numbers. I didn't show you or make a claim about those numbers because I don't know how to do that honestly, and I don't think it's what these sorts of models are for. This is the only model that's ever been proposed for how you could get this level of complexity so early on in life. The only other alternative that I know of is panspermia, which just says that life came from another planet. Okay, so and then of course you have to say, well, how did life evolve on that planet? And so that's not really a good solution. So there really isn't. So I don't know how we would be able to make this sort of model more specific with real numbers so that you could estimate, yes, this could happen in 800 million years. I'd like to be able to do that, but I think it's a stretch, so I didn't try to do that.
One more question, and we should probably wrap up after that. So you can choose.
I don't mind. You put your hand up first, sir.
I have a question about
About what?
about if you compare the biological system with the polymers, which is without ability to self-reproducing. So if you compare D molecules, it can replicate and generate D molecules. But like the polymers, which is like organic polymers, it cannot reproduce. So if the early molecules, they gradually acquire that ability to replicate itself, and when the more biological molecules come in, they start to form a kind of very simple prototype life.
Mm-hmm.
So
it's some sort of autocatalytic process.
yeah. So if we compare this process, if we think about the robots, is it possible robots gradually learn to reproduce itself and learn to use different components, because there's a lot of components running.
Right.
So what I want to ask is, if you compare these two different processes, they have similarity, or there are some basic principles are different?
So I'm not really sure of the answer to your question. The person who would know about robots is Todd Lipson at Cornell, who tries to make robots that make other robots. But I don't know that the sort of fairy tale that life started, there were some chemicals, some polymers, they somehow got more and more complicated, then they could do autocatalysis, and then life started. I don't believe that. The reason I don't believe it is this: if you look at the core of metabolism, the basic core of metabolism is the TCA cycle, the reverse citric acid cycle. And people say, well, all you've got to do is find life that can find all the steps, the 20 steps, or whatever it is of this cycle, and then it can start to be autocatalytic. And I think it's very unlikely that that happened like this. I think it's more likely that life was actually modulated by external periodic perturbations, like the day-night cycle, or convection in a hydrothermal vent, or something like this. And it was driven, and the driving force on a partial network, say like a pathway, like the acetyl-CoA pathway or some other important pathway, those gradually drove the production of metabolites that then started to form complexes and eventually, after being driven externally, the system was able to become autocatalytic. So I think So maybe in robots it's like that. The robots still need to be driven externally. You need professors of engineering and computer science to keep making the robots, but maybe eventually you can make ones that can make themselves. So I think there is an analogy like that, but I can only give that kind of answer.
All right, well, let's thank Nigel.